Hello, I'm Samuel Wagner, and I'm Professor of Infection Biology at the Medical Faculty of the University of Tübingen, and I'm heading the section for Cellular and Molecular Microbiology at the Institute of Medical Microbiology. My lab overall investigates bacterial type 3 secretion systems that inject proteins into eukaryotic host cells. In my lab, we investigate the assembly, structure, and function of bacterial secretion systems, very complex several megadalton secretion systems that gram-negative bacteria and pathogens in particular use to inject proteins into eukaryotic host cells. And Salmonella, for example, uses a type 3 secretion system to inject proteins into cells of the intestinal lining. And these systems help Salmonella to invade into this tissue and also to survive intracellularly. So these systems are really important for the establishment of infection, which in the end also leads to diarrhea, for example. We also work on the type 4 secretion system of Legionella that helps Legionella to survive inside macrophages, which ultimately leads to pneumonia. In addition, we seek to identify small molecule inhibitors against these secretion systems and to develop them as so-called pathogenicity blockers. These very specific inhibitors could in the future be ideal replacements for antibiotics, since they specifically target the pathogens and leave the beneficial microbiota intact. To analyze secretion of these secretion systems, we recently developed a luciferase-based tool that is highly sensitive and allows us not only to monitor secretion even over time and the kinetics of secretion, but even the injection of proteins into eukaryotic host cells. If we combine this secretion analysis with bacterial genetics with different knockout mutants, we can really study the assembly and the function of these systems in detail. To study the assembly of these secretion systems, we developed a genetic tool that allows us to deliberately switch on and off the expression of individual components of the system. And in this way, we can really finally follow the critical assembly steps of, for example, type 3 secretion systems in Salmonella. Analysis of the assembly actually happens by mainly two different techniques. On the one hand, we use the so-called in vivo photocross-linking, where we encode artificial amino acids into specific sites of the proteins that interest us. And these artificial amino acids can cross-link to very nearby neighbors upon UV irradiation, and in this way capture an interaction. We can follow this interaction then by Western blotting or mass spectrometry and also in different mutants, and deduce a very detailed assembly pathway. We also use Blue Native page of purified membranes to look at the entire complexes and look at the dominance of individual complexes in different mutants. And together these techniques are really powerful to provide an overall picture of assembly of these multi-megadalton complexes. To study the continued targeting and biogenesis of injected membrane proteins, we also use fluorescence microscopy, cell fractionation, protein-protein interaction analysis, and cell culture in these cell culture infection models. Last but not least, we develop high-throughput screens to identify new inhibitors of these secretion systems, and we develop these active compounds that we identify into hit and lead structures. And this we do in very close collaboration with structural biologists, with experts in computational drug discovery, and with medicinal chemists. I would like to highlight three overarching projects of the lab. We were the first to isolate the core export apparatus of these Salmonella type 3 secretion systems. Together with our collaborator Susan Lee, at the time at Oxford University, now at the NIH in the US, we were able to solve the structure of this complex that is composed of 4 different components and 11 subunits in total. The surprising part here was that all these 4 proteins are predicted to be integral in the membrane proteins of the bacterium. What we found was that they actually assemble into a helical structure that in the end is not in the membrane anymore, but sits in this base complex of the type 3 secretion system above the membrane. And this helical nature of this complex is so important because it templates the further assembly of the needle structure of these type 3 secretion systems, and this needle in the end reaches to the host cell and is really important to inject proteins into the host cell. One major goal of our lab is to identify small molecule inhibitors of these secretion and injection systems. And in the last years, we were able to identify a small molecule that blocks not the secretion of the Salmonella type 3 secretion system 1 itself, but that actually blocks the major transcription factor HILD and its activity. Since this transcription factor is blocked with this compound, none of the genes of this type 3 secretion system is made if we treat the bacteria, and this renders overall the bacteria essentially non-infective. Using genetic, biophysical, and structural biology techniques, and also medicine and chemistry, we were able to identify the exact binding site of this inhibitor to the transcription factor and perform also a deep structure-activity relationship analysis that helps us to reach compounds with a higher activity and develop them into a lead structure for further drug development. Yeah, one fascinating aspect of these secretion systems is their ability not only to secrete soluble proteins from the cytoplasm into the cytoplasm of the host cell, but to also secrete hydrophobic transmembrane proteins that are destined to be inserted into membranes of the eukaryotic host. This creates actually a problem inside the bacterium because hydrophobic membrane proteins are usually co-translationally inserted into the inner bacterial membrane. In case of type 3 secretion systems, these membrane-inserted proteins would be lost because the type 3 secretion system can only accept substrates from the cytoplasmic side. We identified two mechanisms that type 3 secretion systems use to prevent this wrong targeting and ensure the correct direct targeting to the system. On the one hand, these substrates have a reduced hydrophobicity. This is just so much reduced that the wrong targeting cannot take place, but that they are still sufficiently hydrophobic to insert into membranes of the eukaryotic host. On the other hand, more hydrophobic substrates are actually protected by specific type 3 secretion chaperones that make sure that this wrong targeting cannot take place, but that these proteins are directly fed into the type 3 secretion system. For type 4 secretion systems, like the one we study in Legionella, the situation is a little bit different. Here we realized that these transmembrane proteins are actually secreted in a unique two-step secretion pathway, where they first insert into the bacterial inner membrane, and then they get extracted from the bacterial inner membrane towards the cytoplasmic site, most likely by the coupling complex of the system, to then be secreted by the complex into the eukaryotic target cell. The relevance of our research comes from the pathogenic potential of the bacteria that we investigate. Salmonella, for example, are a major gastrointestinal pathogen, a global burden, not so much in Germany and in Western countries, but in particular in Africa, for example, or in Southeast Asia. And several million people get infected with this pathogen annually, in particular children. So developing novel anti-infective strategies that would be able to complement or replace antibiotics is really an important strategy to overcome antibiotic resistance in the future. This is important because it spares the beneficial microbiota and is so specifically targeted only against pathogens. And this is exactly what we aim at globally in the Cluster of Excellence. To be able to develop new pathogenicity blockers, we do not only need to focus on the direct development of small molecules that inhibit these type 3 secretion systems or type 4 secretion systems, but we also need to investigate into basic research to increase our basic or mechanistic understanding of the secretion function of these systems. So an investment into basic research is also really important for the future. More sophisticated analytical and genetic tools and artificial intelligence-based structure prediction and modeling will really bring us further for the development of new drugs in the future.